Hello friends, welcome to Viologia EXtrema, where we teach biology from the basics with extreme. Myself Keshav Bansal, and today we are going to study glycolipids. Okay, so what these glycolipids are, we know from the term that it is a type of lipid. Which type of lipid and where it is found? So it is a membrane lipid. Okay, it is a membrane lipid, and we have already discussed that membrane lipids can be of three types majorly. Okay, they can be a phospholipid, they can be a glycolipids, today's topic, they can be a sterol. Okay, and we have already discussed this phospholipid in a much detail. Okay, you can check out the video, and these are more important ones because especially when we are talking about animal cell, so the phospholipids are the most abundant type of membrane lipid. Okay, so phospholipids are very important, and also it can be asked in the MCQs in the exams too. Okay, which type of membrane lipid is most abundant? So phospholipid is abundant, and specifically they can ask you that which type of phospholipid. Okay, even in the phospholipids there is phosphatidylcholine, which is the most abundant phospholipid. Okay, or you can say a membrane lipid when we are talking about an animal cell. Clear? So this is important. So as we know, as we studied in the phospholipid, okay, then there is a lipid, we know that it is a lipid, and then there is a phosphoterm, okay, which represents that there is a phosphate in the lipid. Okay, so that is constant. And when we studied the phospholipid, we have seen different types of phospholipids. So there is one thing that constant in that it has a lipid molecule and there is phosphorus in it. Okay, so same here. Okay, it is a lipid, and as the name suggests that there is some glyco thing that is constant in it, and that glyco is referring to as carbohydrate. Okay, you can also call it as saccharide group. Okay, that can be a monosaccharide like glucose, galactose. Okay, that can be a disaccharide, that can be a disaccharide like lactose. Okay, and then it can be more basically, but they can be usually a monosaccharide or they can be a disaccharide. Okay, in this lipid stuff. Claro? So let's look at the structure. I've already drawn it, okay, during the preparation. So basically it will save you a lot of time. Okay, so basically we have alcohol. Okay, we have alcohol. Even in the phospholipid, what we did, that we have alcohol, and to that alcohol we have a fatty acid attached to it. Okay, it can be a single fatty acid or it can be more than one. Okay, that we will see. So alcohol to alcohol, a fatty acid is attached. Okay, this much makes a ester, and the main part, instead of having phosphorus, because the phospholipid there is phosphorus compulsory. Okay, so but here because it's glycolipid, the carbohydrate part is compulsory. So whichever glycolipid we are studying, it is going to have carbohydrate moiety in it. Okay, because that makes it a glycolipid. Clear? So we have alcohol, that alcohol, fatty acid can be attached, fatty acid is attached, and a carbohydrate is attached. Okay, the fatty acid can be it can be one or it can be two fatty acids. Okay, molecule, and the carbohydrates, it can be monosaccharide, it can be disaccharide. Basically we use the term mono and oligosaccharide. Okay, but the examples we are going to use, they contain only monosaccharides and disaccharide. Clear? And the main thing here is this alcohol part. Okay, the alcohol part. So the alcohol can be glycerol or it can be sphingosine. Okay, the alcohol, it can be, wait a bit. Okay, the alcohol, it can be glycerol, okay, or it can be sphingosine. Based on this, we are going to make two types of the glycolipids. Okay, first start with the sphingosine alcohol. Okay, it's the same thing like when we studied phospholipids, we classified phospholipids into two categories based on the alcohol. Okay, we made glycerophospholipids, or we can say phosphoglycerides, and we made because of the sphingosine alcohol, we call them sphingophospholipid. Okay, so sphingolipids can be a phospholipid too, and a sphingolipid can be a glycolipid too. In this case, we are having sphingosine alcohol, so we can call it glycosphingolipid. Okay, so let's first start with the glycosphingolipids. Okay, so basically we know that the alcohol is a sphingosine alcohol. Okay, and to that a single fatty acid is attached, and to that a carbohydrate is attached. Okay, that carbohydrate, and what we know is the same thing in the phospholipid, that this sphingosine part, okay, the sphingosine part when attached with the fatty acid, okay, this region, okay, this is called ceramide. Okay, when a fatty acid is attached to an alcohol, we can refer to that component as ceramide. So either I can say that sphingosine and fatty acid, to this complex a carbohydrate is attached, name the lipid, it is a glycolipid, and it is a glycosphingolipid specifically. Or either I can ask you that to a ceramide group I have added a monosaccharide or a disaccharide, which type of lipid is it? Okay, so then again it's referring to as glycolipid, and specifically as glycosphingolipid because of the alcohol. Claro? So ceramide component is this sphingosine plus fatty acid. Claro? Now the carbohydrate, it can be a monosaccharide. Okay, the carbohydrate can be a monosaccharide, or it can be a disaccharide. Okay, monosaccharide or disaccharide. So let's start with the monosaccharide one. Okay, if the monosaccharide is glucose, or it can be galactose. Okay, if we are using a monosaccharide, basically, then we are getting a cerebroside. Okay, that in this case we are used two examples, glucose or galactose. Clear? And if we are using disaccharide, as a carbohydrate moiety, then, or we can specifically say that if we are using oligosaccharide, okay, because oligosaccharide can have more than one saccharide units attached to it. So it can be di, it can be tri, it can be tetra, and so on. Okay, so here the examples which we have used is the disaccharide. So basically disaccharide is a type of oligosaccharide. Okay, so disaccharide is a which disaccharide we use in the example, we use lactose. Okay, so when we are using a disaccharide, then we are using a we are referring that sphingoglycolipids or glycosphingolipid as globoside. Okay, now both of these cerebroside and globoside are neutral lipids. Okay, why these are called neutral lipids? Because at pH 7, okay, they have zero charge, they are neutral. Clear? But what happen if we take a globoside, and we add an acetyl neuraminic acid. Okay, here it is mentioned wrong. N-acetyl, it's not N-acetyl glucosamine, it's N-acetyl neuraminic acid. Okay, I am editing it here, that instead of glucosamine it's neuraminic acid, N-acetyl neuraminic acid. Okay, and this N-acetyl neuraminic acid is a type of sialic acid basically. Okay, so we are adding, if we add a sialic acid to this globoside, then we are getting a ganglioside. Okay, and previously we have said that these globoside and cerebroside are called neutral lipids because at pH 7 they are neutral, they are uncharged. Okay, but due to the addition of the sialic acid, now this globoside is called ganglioside, and it is negatively charged. It has acquired, now you can guess that from which part this negative charge is coming. So it has acquired this negative charge from the sialic acid basically. Okay, so I think this much is clear. Okay, now we'll look at the notes. Okay, we'll take a read. So lipids plus saccharide. Okay, saccharide is referring to this carbohydrate group, and we know one thing, that as we know that the plasma membrane is a lipid bilayer, and when you have a bilayer, there are two different sides. Okay, one is facing the extracellular fluid, ECF, then I can refer to it as outer leaflet. because it is outer to the cell. Okay, and this one is facing ICF, inner cellular fluid, or we can say the cytosol. Okay, cytoplasm. So this is inner leaflet. Okay, so what happened that the carbohydrate stuff now, this is present in the outer leaflet only. Okay, so whether these carbohydrate making any complex with the lipid molecule making glycolipids, or making any complex with the protein forming glycoprotein, so they are going to be present in outer leaflet only. Okay, this is very important. Clear? Because carbohydrate can be only present in the outer leaflet, so that's why glycolipids also occur in the outer leaflet. Okay, this can also be asked in the exams too. So and okay, and it is a type of membrane lipid. Clear? Then before discussing the types which we already discussed, okay, one thing is very important, that what is the function of these glycolipids. Okay, so what happened due to the presence of this carbohydrate moiety in it, okay, they can help in the determination of blood group. Okay, now without going to much detail into the how these different blood groups are determined, basically you can look that they are A blood group, you know that there B, and there can be O blood group. Okay, what happens that H antigen is going to be compulsory. Okay, so in O blood group, let's start from the O blood group, H antigen is going to be present. Okay, now in the A blood group, H antigen, there is some modification in the H antigen. Okay, which forms A antigen. Okay, from the H antigen, by adding some carbohydrate, some more carbohydrate, okay, we are getting A antigen. Clear? And when we add some other carbohydrate stuff to the H antigen, okay, to the H antigen molecule, then we are getting B antigen. So basically all these three blood groups are determined by the same antigen, H antigen. Okay, in A there is some modification, and B there is some another modification. Okay, and that modification is in the form of carbohydrate stuff. Okay, that we have added. Okay, so they're distinguished by the carbohydrate in them. So that's why we are saying that the glycosphingolipids are determinant of blood groups. Okay, because due to that glyco part. Okay, now what we studied, that the glycosphingolipids, okay, they can be divided into one more thing that we have studied, that they can be glyceroglycolipids. Oh, okay, and they can be sphingoglycolipids based on this. So we are studying first the sphingoglycolipids. Okay, so glycosphingolipids, okay, they do not contain any phosphate, obviously. Okay, phosphate is not going to be present in any glycolipid. Okay, if there is phospholipid, then it's a phospholipid. We have a different category for it. We have studied Cerebroside, Globoside, or Ganglioside. Okay, so in the Cerebroside, I have told you that there is going to be a single monosaccharide unit. Okay, that monosaccharide can be, for example, in this case we are using a galactose, we can use a glucose. Okay, if we are using a glucose, we can say glucosyl cerebroside. If we are using a galactose, we can say galactosyl cerebroside. One important difference is that if we are using a galactose sugar moiety, a monosaccharide, then in that case these type of basically, so galactosyl cerebroside are more found in the neural tissue, this type of glycolipids. Okay, in the membranes of neural tissue, neurons basically. Okay, so and if we are using glucose, so this type of cerebroside, glucosyl cerebroside, these are found in non-neural tissues. Okay, so this is a important distinction. If we talk about globoside, okay, if we add an oligosaccharide, okay, that can be include di, tri, tetra, and so on. Okay, here we are using a disaccharide, lactose, so lactosyl ceramide. Okay, and both of these, cerebroside and globosides, are neutral lipids. Okay, because they have no charge at pH 7. But if I do some modification in this globoside, as we have talked about earlier, then it become a ganglioside. What modification we did? We add an acetyl neuraminic acid. Okay, neuraminic is important. Okay, and it is a type of sialic acid. Now due to addition of this sialic acid, it acquires a negative charge at pH 7, so it is no longer a neutral lipid. So, okay, so ganglioside is basically a modification of globoside, okay, and it distinguishes it from the globoside. The example we use is a ganglioside GM2, okay, and it has other functions like obviously it is going to be outer leaflet, okay, because all the glycolipids due to the glyco part, due to the carbohydrate, they are going to be present in the outer leaflet only. So it helps in the recognition of extracellular molecules which are present on the surface or neighboring cells, okay, surface of neighboring cells, or any extracellular molecule in the recognition of it. So basically this we have studied the glycosphingolipids. Now we will start with the other part, okay. So here, because you will get the notes, okay, in the description, and here they mentioned as galactolipids or sulfolipids, and here they kind of created a confusion, okay, that the galactolipids and sulfolipids are the same thing, okay. We are getting just this structure only, okay, but we will look at it like I said that they can be glycosphingolipids due to this sphingosine alcohol, and in case due to the different alcohol group, the glycerol group, they can called as glyceroglycolipids. That term is more correct, okay, and in other books it is referred to as glyceroglycolipids. This image is from Leninger, okay. So in Leninger they kind of created this confusion by by putting specific examples here, okay, and giving a common structure to it. Clear? So we can, instead of here writing it is at galacto or sulfo, we can write glyceroglycolipids. It's the same case with the phospholipid. We have used glycerophospholipid and sphingophospholipid, okay. So here we are using glycosphingolipid and glyceroglycolipids. So let's look at the structure of glyceroglycolipids, okay. So basically where is the, yeah, okay. So in the sphingophospholipid, what we have seen not sphingophospholipid, in the glycosphingolipids we have seen that there is sphingosine alcohol, and to that sphingosine alcohol there is only one fatty acid attached to it, okay. But here in the glyceroglycolipids, what we will found that is a glycerol alcohol, okay, there is a glycerol alcohol, and to this glycerol alcohol we have two fatty acid attached to it. So we can call this structure as diacyl glycerol, okay. We can call this structure as diacyl glycerol, because diacyl for these two fatty acids and glycerol for this glycerol molecule. So this diacyl glycerol is going to be common, okay, as we will see in the examples. So to this, because that is a glycolipids, so carbohydrate part is compulsory, okay. That carbohydrate, it can be a monosaccharide, it can be a disaccharide, okay. The example which we have used, okay, they contain monosaccharide units. Now that a monosaccharide unit can be in number one, basically there can be one monosaccharide unit, or there can be more than one monosaccharide unit, for example 2 monosaccharide units, okay, but it is not a disaccharide, okay. So, and also one more thing, that and it is not compulsory, okay. From this structure we think that the sulfur group, okay, it is going to be compulsory, but it is not compulsory. Clear? Thus they can be sulfonated too. The carbohydrate can be sulfonated too, okay. So let's start. So this structure is basically called glyceroglycolipids. Now if we go specifically, okay, let's say this is our glycerol molecule. This is a glycerol molecule, to this two fatty acids are attached, so we are referring to as diacyl glycerol, okay. Now if I add I need to add a carbohydrate, okay. So if I add a galactose type of carbohydrate, okay, galactose carbohydrate, it's a monosaccharide, okay, and if I add just one monosaccharide, one galactose residue to it, okay, specifically galactose residue, okay, so then it becomes monogalactosyl, okay, galactosyl for the galactose, so monogalactosyl, and we know that we use diacyl glycerol for the rest of the complex, diacyl glycerol. So monogalactosyl diacyl glycerol we can refer to as MGDG, monogalactosyl diacyl glycerol, okay. And if I use two galactose molecule, then I put the term di instead of mono, then it is digalactosyl diacylglycerol. Okay, so this is basically these are called, due to this galactose, these are called galactolipids. Okay, galactolipids, and these are type of glyceroglycolipids. Claro? Now if we make some changes, if we are using the same diacylglycerol component, and now instead of using galactose as a carbohydrate, we are using glucose as a carbohydrate, and that glucose is sulfonated. Okay, that glucose is sulfonated. So now we are referring this type of glyceroglycolipids to as sulfolipids. Okay, because the carbohydrate moiety is sulfonated in it here. Okay, so let's look at the notes. So the galactolipids, they consist of glycerol, fatty acid, galactose residues. They can be one, they can be more than one. Important thing is that, I've just told you in the beginning that the major three types of membrane lipids can be phospholipid, they can be glycolipids, okay, or they can be sterols. Okay, and if I talk about the animal cell, in case of animal cell, the most abundant type of phospholipid, in case of even other bacteria stuff too, they can be phospholipid. Okay, the abundant is phospholipid, okay, type of membrane lipid. But in the case of plants, in the case of vascular plants, because they may have a deficiency of phosphorus, so kind of plants are smart enough to modify their major membrane lipids to be glycolipids. So because phosphate is not present in here, okay, so they made this modification due to the deficiency, because plants usually face the deficiency of phosphorus in the soil. Okay, so instead of using phosphorus for making the membrane lipids, they utilize for other stuff only. So here the vascular plants contain 70 to 80% of membrane lipid is this galactolipids. Okay, so which is a glycolipid basically, and they are specifically present in the thylakoid membrane of the chloroplast. If you don't know the chloroplast structure, the basic is that this is a chloroplast molecule, is cell. Okay, so the chloroplast, we have stroma, okay, and we have these grana. Okay, these grana of thylakoids. So basically these grana consist of many grana, or we can say many thylakoids. Okay, so the membrane of this thylakoid, they contain this galactolipids. Okay, and these two examples we have already studied: monogalactosyl diacylglycerol or digalactosyl diacylglycerol. And if the carbohydrate moiety is a glucose residue and it is sulfonated, so it is sulfonated glucose residue, okay, which is attached to a diacylglycerol molecule, then we refer to them as sulfolipids. These are also found in the plant membranes. Clear? So this is all about the glycolipids. If you understand the video, then like the video, share it and subscribe our channel. Okay, and you can help us in this noble service of providing free notes and quality lectures by donating a small amount. Okay, even a small help from your side creates a huge difference. So thank you from Viologia EXtrema. See you next time. Take care.